The first process of gene expression is transcription, which occurs in the nucleus. Transcription occurs in three stages: initiation, elongation, and termination. On the DNA strand, a gene begins with an initiation site and ends in a terminator site. During initiation, an RNA polymerase enzyme binds onto the initiation site of a DNA strand.
During elongation, the RNA polymerase unzips the double helix structure and slides along the DNA strand. As the complementary base pairs pair up, the RNA polymerase binds nucleotides to the 3' end of the newly formed RNA strand. Nucleotides contain a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen bases are adenine, thymine, guanine, and cytosine. The order of the four bases will determine the instructions or genetic code. When the RNA polymerase reaches the terminator site, the polymerase disbands from the DNA strand and releases the messenger RNA into the nucleus.
When the mRNA is released from the polymerase, it is pre-RNA, before it's processed and edited and becomes mature mRNA.
Splicing removes introns, since they are non-coding sequences that must be removed before translation, therefore only leaves exons. Spliceosomes splice and pinch the introns out and connect the two exons together, therefore leaving it as mature RNA strand.
The mature mRNA strand leaves the nucleus and enters the cytoplasm, where the process translation can begin. The nitrogenous bases are grouped into triplets called codons. A small ribosomal subunit binds to the start codon of the mRNA strand as a tRNA molecule binds to the start codon with an anticodon.
Once the initiator tRNA binds to the start codon, a large ribosomal subunit binds to form a translation complex, which has three sites, E, P, and A. The tRNA molecule carries an amino acid, and the codons on the mRNA strand ensure that the correct amino acid is brought into the A site. The initiator tRNA moves to the E site and leaves the amino acid with the previous tRNA, which has entered the A site. As this process continues and each tRNA leaves its amino acids, the chain of amino acids grow, creating a polypeptide. Once the ribosome hits the stop codon, a release factor is brought in to end the process. Once the release factor enters the ribosome, the polypeptide chain is released and the ribosome and the tRNA dissociate. After dissociation, the polypeptide may need to be modified. These modifications occur in the rough endoplasmic reticulum.
Protein synthesis.
Oh, and we forgot one thing.
When DNA is transcribed, errors occur which result in mutations.